What happens after a medicine reaches real patients and rare harms begin to appear? Pharmacovigilance exists to detect, assess, understand, and prevent adverse effects across the life of a product. In this video, you will trace why this work became essential, how cases are handled, and who keeps the system running. The World Health Organization defines pharmacovigilance as the science and activities relating to the detection, assessment, understanding, and prevention of adverse effects. Its scope includes medicines, vaccines, biological products, herbal remedies, and blood products. It applies during clinical development and across the whole marketed life of a product. The aim of pharmacovigilance is not zero risk. It is an informed and continuously updated judgment about benefit against risk in real clinical use. This work turns individual patient reports into evidence that can change labeling, prescribing advice, or market status. And for a marketing authorization holder, it is a legal obligation with inspection consequences. Pharmacovigilance exists because pre-authorization trials cannot reveal everything. Trials usually study a few hundred to a few thousand patients, so rarer reactions are often missed. They also exclude many groups, including children, older people, pregnant women, patients with renal or hepatic impairment, and those taking complex co-medication. Trial follow-up is usually short, so delayed effects such as malignancy, fibrosis, or developmental harm may not appear before authorization. Real practice adds off-label use, medication errors, poor adherence, and drug, food, and herbal interactions. A review identified 462 medicinal products withdrawn worldwide for adverse reactions between 1953 and 2013. In that same review, the median interval between the first reported reaction and the first withdrawal was 6 years. That gap shows why continuous monitoring matters after approval. Pharmacovigilance tries to shorten the distance between the first warning sign and a decision that protects patients. Thalidomide was marketed from the late 1950s as a sedative and antiemetic, including for morning sickness in pregnancy. It caused severe limb and organ birth defects, and no systematic reporting system existed to detect the pattern quickly. That failure helped create the modern foundations of drug safety regulation. The United Kingdom response included the Committee on Safety of Drugs and, in 1964, the Yellow Card Scheme. The United States passed the Kefauver-Harris Amendments in 1962, requiring proof of efficacy and stronger safety oversight. A World Health Assembly resolution in 1963 led to the World Health Organization Program for International Drug Monitoring in 1968. Practolol caused oculomucocutaneous syndrome. Venoxaprofen, marketed as Opren, was withdrawn after serious adverse reactions in older patients. Later cases showed that safety problems can emerge in different ways. Terfenadine was withdrawn after QT prolongation caused by interaction with drugs that inhibit its metabolism. Cerevastatin, marketed as Baycol, was withdrawn because of rhabdomyolysis. Rofecoxib, marketed as Vioxx, was withdrawn after increased cardiovascular events were reported. Together, these cases show different detection routes, including clinical observation, interaction pharmacology, spontaneous reports, and randomized trial data. An adverse event is any untoward medical occurrence in a patient given a medicinal product, and it does not necessarily have a causal relationship with treatment. An adverse reaction is a noxious and unintended response where a causal relationship is at least suspected. Every adverse reaction is an adverse event, but not every adverse event is an adverse reaction. Under 21 CFR 314.80, an adverse drug experience includes overdose, abuse, withdrawal, and failure of expected pharmacological action. Causality does not need to be proven to report a suspicion. The Medicines and Healthcare Products Regulatory Agency states that suspicion alone is enough. A case is serious if it results in death, is life-threatening, requires or prolongs inpatient hospitalization, causes persistent or significant disability, or is a congenital anomaly. Important medical events also qualify when they jeopardize the patient and may need intervention. Serious is a regulatory classification, not the same as clinical intensity. Unexpected means the reaction is not listed in the current reference safety information, or it is more specific or more severe than the listed event. Expectedness is judged against the summary of product characteristics or the company core safety information in the company core data sheet. These distinctions help drive reporting decisions. The Rawlins-Thompson scheme classifies reactions by mechanism. Type A, or augmented, reactions are dose-related, predictable from pharmacology, common, and usually have low mortality. Type B, or bizarre, reactions are not dose-related, are unpredictable, are rarer, and carry higher mortality. The scheme was later extended to Type C chronic, Type D delayed, Type E end of use, and Type F failure of therapy. This classification helps guide action. Type A reactions often respond to dose reduction, while Type B reactions usually require withdrawal. The Individual Case Safety Report, or ICSR, is the fundamental unit of pharmacovigilance data and the building block of every safety database. A valid case needs 4 minimum criteria: an identifiable reporter, an identifiable patient, a suspect medicinal product, and a suspect adverse reaction. If one is missing, the case is not valid. Follow-up to complete missing information is a core task. Cases can arrive from spontaneous reports, the scientific literature, solicited sources such as patient support programs, clinical trials, and regulatory authorities. The CIO-MSI form is the long-established paper format for reporting individual serious adverse reactions between organizations. Day 0 is the first calendar day on which anyone in the organization receives information that meets the 4 minimum criteria, including weekends and holidays. Triage checks validity, seriousness, and whether expedited timelines apply. Then the case is screened for duplicates. Data entry and coding follow. The reaction is coded in MEDRA, and the medicinal products are coded in WHO drug. Medical review assigns seriousness, expectedness against the reference safety information and a causality assessment, then identifies follow-up questions. In the European Economic Area, serious cases go to EUDRA vigilance within 15 calendar days, and non-serious cases within 90 days. MEDRA standardizes how reactions, indications, and medical history are coded across the industry. It has 5 levels, from system organ class down to lowest level term, and 2 versions are released each year in March and September. ICH E2B governs electronic transmission of ICSRs, with E2B R3 built on the HL7 message standard. Spontaneous reporting is inexpensive, covers all products and all patients continuously, and has no upper limit on observation time. It is the only practical method for detecting very rare reactions. But underreporting is universal. There is no denominator, and reporting can be distorted by Weber effect, notoriety bias, stimulated reporting, and channeling bias. In the United Kingdom, The Medicines and Healthcare Products Regulatory Agency runs the yellow card scheme, and the inverted black triangle marks a medicine under additional monitoring. In the European Union, requirements are set out in good pharmacovigilance practices, and the Pharmacovigilance Risk Assessment Committee assesses safety issues. Patients, healthcare professionals, marketing authorization holders, and global programs all contribute to outputs such as risk management plans, periodic safety update reports, and safety communications. Now you have seen why pharmacovigilance exists, how modern regulation was shaped, and how safety reports move from first receipt to regulatory submission. You have also covered the key terms, the strengths and limits of spontaneous reporting, and the main systems and stakeholders involved. The central idea is simple. Drug safety depends on turning individual reports into evidence that protects patients.